Good afternoon, everyone, and welcome to a second webinar in our series on molecular epidemiology and sequencing approaches in public health. Just a reminder before we get started that this webinar will be recorded. And having some technical challenges here quickly. Yes, so this webinar will be recorded. The format is a 35 to 40-minute presentation, followed by 10 to 15 minutes Q&A. Just a few quick overview on this series. So this is an online training on molecular epidemiology, including application of whole genome sequencing in public health setting. It's a collaboration between the CDC and the CDC Integrated Food Safety Centers of Excellence. And the target audience for this for these modules and this activity are health departments as well as agriculture department staff who use WGS data for foodborne disease surveillance and outbreak investigations, but importantly also those working in other areas that are making use of genomics and next generation sequencing for disease surveillance. The overall program includes 4 online modules that are available on our website, as well as 4 webinars, and this is the second webinar in this series. Here are the titles of the 4 webinars. The first one has already been posted and is available for people to watch. This is number 2, and then 3 and 4 are being scheduled for the coming month, and we'll provide you with more detail on those. The modules are listed here. These are short, about 20-minute modules that provide background that goes along with these webinars. And again, these are available on our web pages. The next webinar after this one, or the webinar today, next webinar after this one will focus on high-quality Snip, whole genome MLST, and the whole genome sequencing alphabet soup, where we focus on advantages of whole genome MLST and high-quality SNP trees, why and how same raw data can yield different trees, and how to interpret trees and other data representation. And I will present this webinar, which will be on May 24th from 3 to 4 p.m. Eastern Time. With that, I want to introduce today's presenter, Dr. Madhu Anand. She obtained a Dr. PhD from the State University of New York at Albany. She has 19 years of experience as an epidemiologist at both the local and state levels. And for the past 16 years, she has worked at the New York State Department of Health, Bureau of Communicable Disease Control in a variety of roles. She currently serves as the Deputy Director of the Regional Epidemiology and Investigations Program. Her main areas of research have focused on foodborne disease surveillance and outbreak investigation. I'm going to turn over my screen to Madhu to start this webinar.
Great. Can you hear me okay?
Yes, we can hear you on your screen is up, so you're ready to go.
All right. So good afternoon. As Martin mentioned, my name is Madhu Anand, and I'll be presenting on how next generation sequencing data are analyzed from raw data to sequence contigs. Before I start presenting, I wanted to thank CDC, Martin Weidman, and my other Centers of Excellence colleagues for a number of the slides I will be presenting today. After this presentation, we hope that you will understand the process of obtaining a contiguous sequence. We also hope that you will appreciate the varied methods of conducting next-generation sequencing, or NGS, and understand how there are differences between conducting NGS for viral versus bacterial pathogens. As we describe how to put contigs together, we will talk about how to do that in 2 different ways, through reference and de novo assemblies. Finally, although we will not go into the nitty-gritty of quantitative measures, you should understand that there are quantitative measures to assess the quality of sequencing data. In the first webinar, it was discussed that what the NGS process is overall, and I wanted to quickly review. You have an isolate that undergoes some agitation, and it's broken up into fragments. From these fragments, you develop what are called sequence reads. The sequence reads are then assembled into contiguous reads, and contiguous reads can then be put together in order. Technically, all put together, it's called a scaffold. And finally, they're compared to each other, to other contiguous reads that are of known etiologies, or sometimes just to see if they are the same or different without knowing the etiology. As an epidemiologist at the state level, I first started hearing of next-generation sequencing and whole genome sequencing about 5 years ago. Local health departments in the area began asking me what the terms meant. It's not until recently that I started to get a better handle on the nuances of them. So the father or mother of sequencing is called Sanger sequencing, meaning it's the first generation. It is a DNA sequencing technology that has been used for approximately 25 years. This has given way to next-generation sequencing, also known as high-throughput sequencing. Next-gen sequencing is an umbrella that includes many different platforms. These platforms reside under either the short read variety or the long read variety. We usually consider the short reads to be the second generation and the long reads to be the third generation. Under the second generation, we have 3 different platforms. The platforms that have been introduced by Illumina, the Ion Torrent platform, and 454 Pyro sequencing. In the third generation, there are 2 different platforms, the Pacific Bio or PacBio, and the Oxford Nanopore. Both Sanger and next-gen sequencing allow for whole genome sequencing. They're just different technologies for doing it. There are several considerations when deciding with which platform to sequence. All technologies can do the job, but there is a balance based on a variety of factors. Cost is a factor when you start looking at switching platforms. Because Sanger has been well adopted, there is less startup cost than if you start incorporating a new technology. With Sanger sequencing, you can sequence a small amount, but the quality is much higher. The sequence read length is another differentiator between Sanger sequencing and next-gen sequencing for most platforms. The PacBio or the Oxford Nanopore both produce long reads, but they come at a price. So with Sanger sequencing, you can obtain reads of about 700 to 800 nucleotides, which are longer reads than you can get with Illumina, but shorter reads than you can get with PacBio. With next-gen sequencing, you have massively parallel sequencing occurring at one specific moment. Millions of fragments are sequenced in a single run, versus with Sanger sequencing, only one forward and one reverse read occur at one given moment. From this illustration, you can see that depending on the platform, there's great variation in read length, volume of isolates that can be run at one time, and cost. Depending on the question that needs answering and the pathogen being tested, one of these platforms may be preferred over another. Epidemiologists should understand which platform their lab is using as next-gen sequencing is adopted more readily. They should do this in order to understand the limitations of what their laboratory can provide. I think a lot of times epidemiologists ask their lab to get testing done for investigations, sometimes without really understanding the turnaround time or the quality of what they might be getting. So it's important that open communication between the two can be extremely helpful in setting up a program in your department. It can set expectations on how often specimens may be run, guide follow-up questions for an investigation, and develop a workflow. For example, at the New York State Department of Health Wadsworth Center Laboratories, they have a NextSeq where they are running all salmonella, but need to wait until there are 96 isolates for a run. Previously, they were using a MySeq that could be filled up faster and run more often, but was more expensive per read. As epidemiologists, we have a better understanding of how the platform may impact turnaround time depending on the season. I think one other thing to mention here is this is kind of where things are at, but they're constantly evolving. Costs and adoption of technology changes very quickly, so what may be expensive today might not be expensive tomorrow. Additional considerations for platforms are what pathogens are being sequenced. It's important to know the read length the platform can handle in relation to the size of the genome being sequenced. And I'm going to go into some specifics on various genomes. Viral genomes are relatively compact at about 10,000 nucleotides and have little wasted space. They're variable in composition, meaning they can consist of DNA or RNA, can be single-stranded or double-stranded, they can be linear or circular, or be single or segmented. They are often highly variable, meaning there are a lot of single nucleotide polymorphisms between viruses. They evolve so fast that you can get different strains very quickly. This is particularly true of single-stranded RNA viruses. Here's an example of a viral genome, hepatitis B. There are a variety of genes in a circular fashion. Each gene is a different length. The genes are coded in different directions, and multiple segments make up the genome. Bacterial genomes, on the other hand, are larger, usually about 5 million nucleotides, and have a highly conserved core portion of about 3,000 to 5,000 genes that are present in most strains of a given species. There is also an accessory genome, which is made up of up to thousands of genes which are not always present. So what are accessory genes' function? They can still be very important for instance, in antibiotic resistance, such as beta-lactam resistance. The structure of the bacterial genome is double-stranded DNA that is usually a single circular chromosome. Complications occur in bacterial genomes with plasmids and phages. Plasmids are circular, double-stranded DNA that can replicate independently from the chromosome. They often carry resistance or virulence genes and can be passed from one bacterium to another. Phages are viruses that can infect bacteria, and the genome can integrate in the chromosome. There are also other repetitive elements that are hard to analyze because we don't necessarily know where they go or how many times they are repeated. They are important because they can cause difficulty in putting together an assembly. Here's an illustration of an E. coli 0157H7 genome. You can see that there are phages and plasmids that are mobile elements that are not typically part of the core. Those are those accessory pieces. Eukaryotes, on the other hand, are huge. They're billions of nucleotides large. The human genome is 3 billion nucleotides, so that can give you some perspective. The structure of eukaryotes is a double-stranded DNA with multiple chromosomes. Most of the human genome has no apparent function, but they are composed predominantly of non-coding segments such as introns, rather than coding segments such as exons. In general, these are the differences in size between viral, bacterial, and eukaryotic organisms. Viral genomes are compact with efficient organization and are the smallest. Bacterial genomes are more complex with repetitive elements and are larger, and eukaryotic genomes are huge and mostly introns. This illustration gives you an idea of relative genome size for a number of entities. Humans fall into about 3 billion nucleotides, and just to give you some perspective on that, Listeria is 3 million nucleotides, so humans are quite a factor more than that. So now I'm going to talk for a few minutes about mobile genetic elements, which I've begun referring to just a little bit now. Mobile genetic elements move around and within and between genomes. They enable what's called horizontal gene transfer and consequently are an important factor in acquired virulence, antimicrobial resistance, and bacterial evolution. Mobile genetic elements are important for epidemiologists to understand as they can allow two organisms to become different very quickly and cause resistance. because they can add from a few thousand nucleotides to potentially hundreds of thousands of nucleotides to a genome, the interpretation of DNA becomes difficult. Some plasmids can be more than 100,000 nucleotides long. For analysis purposes, you need to take these components into consideration and determine whether to keep them in the final analysis. I'm going to talk about some specific mobile genetic elements, the first being plasmids. Plasmids are small extrachromosomal sequences that are circular and self-replicating with a varying copy number. They are present in both bacteria and eukaryotic cells. They have a number of functions, some that are more important for outbreak investigations, such as resistance and virulence. Similar plasmids usually can't coexist. Plasmids play an important role in horizontal gene transfer and play a critical role in bacterial diversity. Bacteriophages are another type of mobile genetic element. They are viruses that infect bacteria and are extremely diverse. They can be composed of single-stranded or double-stranded DNA or RNA. They inject this DNA, RNA into the target bacteria and take control of the bacteria to reproduce. Lysogenic phages are similar in that they do not cause immediate degradation of the cell, but instead incorporate into the host genome. In this instance, the DNA, RNA may remain in a dormant state, but still alter the organism. Transposons are the final mobile genetic element I'm going to speak of. They can mediate their own movement within the genome or between hosts. Transposons are a common feature of most genomes, not just bacteria. They encode an enzyme which cuts the genome at certain segments and can move and insert itself in a different area of the genome. By doing this, they may knock out a gene's original function. This actually isn't used as much in public health. It's actually used more for research purposes right now. So I've talked a bit about the machines needed to do whole genome sequencing and how there might be elements within a raw specimen that need to be taken into consideration for analysis purposes. But in order to understand how an isolate gets to be analyzed, we need to talk about the machinery and the DNA differences of the raw product. But I've not talked about how you put all these pieces together. Before I delve in, let's discuss definitions that are specific to the sequencing process. A sequence is the generic name describing the order of biological letters associated with the DNA, RNA, or amino acids. You will hear the terms reads and contigs many times through this presentation. They are both sequences. Reads are sequenced reads of base pairs as you try to assemble. Contigs are reads that have been assembled together into a final product. Contigs is a term you're going to see a lot later on, and this is simply the long version of it is contiguous sequence of DNA. So why do we need to assemble in the first place? Bacterial genomes, as I mentioned, are millions of nucleotides. Those A, C, T, and Gs, I remember back from college biology. These genomes need to be broken into smaller pieces in order to process and sequence. The current technology looks at pieces of about 250 nucleotides to sequence. However, technology is very quickly evolving. Newer technologies can read greater than 10,000 nucleotides at once. A large percentage of the sequence encodes proteins. The areas where proteins are encoded are called genes. The areas where the proteins are coded need to be aligned so they can be compared to each other. There are two different methods to putting an assembly together. The first is reference guided and is a little easier to explain. So imagine you have a map or a picture of the final product and you're trying to match your pieces together to look like that final product. Let me provide you some concrete examples. You're putting together a jigsaw puzzle and you have the picture of the final product of what the jigsaw puzzle will look like in the cover of the puzzle box. You're putting the pieces together for the puzzle and based on the colors and shapes of the final product image. Another example is that you know the words and composition of a song and are able to put the words of the song into the correct order. So now let's talk about the second version of making an assemblage, de novo. This is where you put the pieces together by what makes sense. Let's take the previous examples. For the jigsaw puzzle, imagine you've lost that picture, the cover of the box of the final product. So are you still able to put the jigsaw puzzle together? Yes, you're able to do that by looking at the shapes of the pieces and seeing what fits together. Likewise, for lyrics of a song, you may not know all or even any of the words of the song, but can figure out sentences from the words that you've been given. Let's walk through de novo assembly of some lyrics. This example indicates each word is a base pair. We put together 4 to 6 words into something that makes sense. In this case, these are the reads. We have 15 lines of reads. We then try to line up all the reads so the words are lining up with each other. When we have them lined up with each other, we can put together a final product, which is the assembled contig. So we have the aligned reads, and now we have the assembled contig. I wanted to mention one more thing. Coverage is also important. As you can see in this middle section, the word right aligns 3 times out of 5, and the word standing aligns 4 times out of 5. A contig developed from reads which have a higher coverage give you a higher confidence that the contig was assembled correctly. However, by putting a contig together through de novo, there is some inherent ability to make errors in reads and then throughout. There are also issues where there are multiple areas in a song with the same lyrics, and you need to figure out where to assemble and how many times. In this example, the words "let it be" repeat many times throughout the song. The number of times they can be put together may be confusing. Is it just once? Is it twice? Is it 3 times? You can either have it in the first instance, let it be, let it be, let it be, or it can keep going on, as you can see in the last example. Once you put together the final assembly, you also might have areas of the song where you didn't have enough confidence they belonged, where you essentially didn't have good coverage, so you wouldn't include them. And when you conduct a reference-guided assembly, for example, when you know that you are looking at a certain Salmonella serotype, you might find some unmapped reads that occur for a variety of reasons. The sequences are not actually in the reference. Plasmids may not be present that are extra chromosomal and not part of the gene, or DNA may be a variant or have rearranged. The advantage of conducting reference-guided assembly is that it is relatively fast and well-suited to highly conserved genomes with little change. The disadvantages include that there can be issues if the genome is highly diverse and if there are lots of mobile elements that might lead to unmapped reads. This is an example for when you conduct a de novo assembly. For example, when you are not sure what pathogen you are looking at. Advantages include that de novo assembles all the reads it can and doesn't eliminate any based on a reference. I won't go into the details, but there are many algorithms out there to be able to do this. The disadvantages include that you do not always get things right, particularly with complex repeats, as we saw earlier. In order to view the sequence information, there are computer programs the lab and bioinformaticians use. I've put up an example of how putting sequence reads together may look. I now wanted to spend a few minutes speaking about how to assess assembly and quality. As an epidemiologist, I have confidence that the clusters that are reported from the laboratory are based on sound methodologies. But what I did not know is that there are tools they can use to give them higher confidence in the accuracy of the reads and contigs that have been assembled. I won't go into all of them, but they have measurements and metrics to assist them in making assemblies based on the size of these contigs, the coverage of the reads, especially in the repeat areas. There are also ways to improve assembly, which include having higher coverage, having paired instead of unpaired reads, and having a longer read length, ensuring read quality, and utilizing assemblers. Specific to read length, as initially discussed in this presentation, read lengths depend on the machines and instruments used. Depending on the goal of assembling, for example, whether to find clusters versus to identify the assembly, one may be better than the other. Shorter reads are usually economic, but may have more repeats and missing areas. Long reads have better coverage, but technology is more expensive. Long reads are typically not used in public health sequencing. For public health, long reads are only used for niche purposes, such as generating high-quality assemblies for validating sequencing protocols. The reason why we don't use long-read sequencing for outbreak surveillance or foodborne pathogens is the cost and comparatively high error rate. This might be overcome by increasing coverage, which in turn would make it even more expensive for most labs. The type of sequencing that is currently relevant is short-read sequencing, and the MiSeq is the instrument most labs are using to achieve this at this point in time. This is an example of a 33-word read. High quality gets the lyric words in order correct. Illumina sequencing has good ability to assemble. However, with poor quality, which you would still have with long reads, you start to see misspellings with words, but still have the general feel and get the correct order. While this looks like it might give you poor quality, the long read sequencing gives you a better ability to put sequence reads in the correct order. This is where the PacBio machinery might come in. For example, take a rare salmonella. I recently heard of Salmonella Livingstone. And you might not already have a good sequence run at this point for Salmonella Livingstone. You can use the PacBio platform to get better coverage, and you can use that to get a genome as a reference against Illumina runs where you can call Snips. The benefit of long reads is you get the order correct. Let's discuss some quality control metrics that are associated with next-gen sequencing instruments. One of the QC metrics that you hear about the most is the Q-score, Fred Quality Score. A Fred Quality Score is a measure of the quality of the identification of the nucleobases generated by automated DNA sequencing. This is something that is universal across all the platforms. A coverage is another quality control metric. The coverage is the average number of reads representing a given nucleotide in the reconstructed sequence. So after the sequences have been aligned to a reference genome, or without a reference genome, known as de novo assembly, you will be able to see how many redundancies there are for a specific base. We call that coverage. If the coverage is minimal, then each base holds less weight than when it comes to calling that specific base. If the coverage is very high, then you know if it's adenine. It would be a high probability that adenine is the correct call. Another quality control metric is the insert size, which is the length of the DNA that you want to sequence and that is inserted between the adapters. One of the other tools that are available are quality scores. This is the likelihood that a base call is correct. Some quality scores are called FREDs, which is where the sequencer scores base call quality based on a control. This is broken out further by the percentage of base calls that have a 1 in X chance or less of being incorrect. For example, Q20 is 1 incorrect in 100 base calls. Q30 is 1 incorrect in 1,000 base calls. Essentially, this helps to determine whether a base call is trustworthy and can be used in a high-quality SNP analysis. Next, let's describe coverage in a little bit more detail. Let's start with the interquartile range, or IQR. This is the difference in sequencing coverage between the 75th and 25th percentiles of the histogram. A high IQR indicates high variation in coverage across the genome, while a low IQR reflects more uniform sequence coverage. Below, in the diagram, you can see that the two different scenarios On the left, you have a smaller IQR, whereas on the right, you have a more diverse or spread out IQR. On the left is ideally what you would like to see across your reads. On the right is a little bit too much variation or a high IQR, which is less desirable. A little bit more information on coverage. The mean or mapped read depth is the sum of the mapped read depths at each reference base position divided by the number of known bases in the reference. A nice formula. The mean read depth metric indicates how many reads on average are less likely to be aligned a given reference base position. Whereas raw read depth, this is the total amount of sequence data produced by the instrument. So it's pre-alignment, and it's divided by the reference genome size. Although the raw read depth is often provided by sequencing instrument vendors as a specification, it doesn't take into account the efficiency of the alignment process. If the large fraction of the raw sequencing reads are discarded during the alignment process, the post-alignment mapping read depth can be significantly smaller than the raw read depth. Finally, the last quality control metric we're going to discuss is insert size. This image here is a great basic image to help show the insert size compared to the adapter regions on either end of the insert. So when you look at the total fragment for next-gen sequencing platforms, you have the total fragment length that includes the adapters and the inserts. Read 1 adapter, read 2 adapter, and then you have the insert size and you have the inner distance or the distance between read 1 and 2. Now, I mentioned many times the term SNP or single nucleotide polymorphism. A SNP is where there is a nucleotide difference in the sequence. Sometimes there are insertions or deletions. They're called indels. They might look like SNP differences, but for the most part, SNPs result in phylogenetically informative differences, whereas indels may not. For analysis that look at high quality SNPs, indels are not taken into consideration. And this is an important point because there are 2 different analyses that can occur. One is SNP, and now I'm going to talk a little bit about whole genome multi-locus sequence typing, or MLST. Whole genome MLST is another method for analysis. There are predefined loci or genes which equate to short reads. For example, with Listeria, there's X number of genes. When looking at just one of those loci, you can take a look at the actual read and look for differences. In this example, the first read is allele 1. When you look at the next read, there are 2 SNP differences. We would call that a different allele and number it sequentially as allele 2. This is not named allele 2 because it has 2 SNP differences, but because it's the next one where we saw a difference. We look at the next read and see that it has 1 indel and 1 SNP and call it allele 3. Notice that when it comes to MLST, we are including indels as a difference from the original read. Now we're going to start to line up those short reads or loci or genes, as I mentioned before. They can all be different sizes. So we have loci 1, and we have 3 different isolates we're looking at. We look at each gene and compare against the same area across the isolates. So for this example, when we are looking at locus 1, isolate A, B, and C are all the same. When we look at locus 2, isolate A and B are the same, but isolate C is different. We take a look to see if the sequence is the same across isolates or different, and if they are different, then they are called a different allele number. In this sense, it doesn't matter if there are 1 or 20 SNP differences within the gene, for example, within locus 1, an isolate at that loci would just be counted as different. So as far as MLST analysis, it can be faster than analyzing for SNP differences. And often what happens is MLST is conducted twice, but you can still get an answer faster. And how it's conducted twice is, first what happens is comparisons are made between allele calls made with the short reads, but then those short reads form a contig, and then comparisons are again made between allele calls with the contig. This eliminates machine noise that is present with either method when done independently. So we're looking at Snip versus MLST analysis, and there's a benefit for both of them. Both analyses are conducted from raw data. The benefit is that when reads are put together, they don't need to be put in order, so that analysis can be quicker with MLST. The con is that this type of analysis may not be able to account for mobile elements, which might be important for antibiotic resistance, etc. But at the end of the day, it's a question of how much does the horizontal gene transfer matter when trying to analyze for outbreak investigation and cluster investigation, and only in specific instances or when you have specific questions is it very critical to go to a SNP analysis. So in summary, the generation of sequence data that will be used by epidemiologists involves both hardware technology as well as fairly complex bioinformatics analyses. Epidemiologists will not have to know the details of the genome assembly and SNP detection process, but they should know that the specifics of the analysis affect conclusions, and they should be prepared to ask questions about sequence data. SNP and MLST analysis both have a place in analysis. The two are not mutually exclusive. You can do both analyses with the same raw data. For some cases, MLST works better, and for others, SNP might work better. In important cases, it makes sense to use both approaches for data analysis. Just one final word. I wanted to mention that learning about whole genome sequencing has been an evolution for me. It's been a process of being exposed to essentially a different language and learning the terminology and how to use it in the right context. And it's actually taken years of hearing the same thing, putting it to practice, re-listening, and getting a more discrete understanding. So I just wanted to mention that everyone listening today may be at different stages in the same path, and I encourage you to come back to not only this presentation, but the other presentations that the Centers of Excellence are doing as you start getting a more stable footing or understanding, because I think the more you hear the same thing, you pick up something a little bit different each time. So I wanted to thank a number of partners in helping me put together this presentation, specifically the Centers for Disease Control and Prevention. I really appreciate the help from Greg Armstrong, Peter Gernerschmidt, and John Besser. Martin Wiedman from Cornell has been invaluable, as well as my colleagues at the Integrated Food Safety Centers of Excellence. And I will leave it for questions.
Great, thank you, Madhu. So we're opening up for questions because we have a large number of attendees. We really can't do live questions where you can talk, but feel free to type them in either the webinar chat or the specific Q&A box, which you can type the questions in, and then we will take them and I'll read them out and then we can answer them. We got a couple of questions about recordings, so just to clarify, we will make available both a recording of the webinar as well as make available the actual slides without the recording. So if you want to use the slides and utilize them for internal training, otherwise you can do that too. So both the slides as well as the recorded webinar will be made available. So with that, as I said, I want to encourage you to type in some questions. To maybe get us stories to think about. I'm going to try to ask one. So Madhu, I mean, obviously you have experience with applying whole genome sequencing in the epidemiology side. So I'm curious, as you interact with the lab, how often do you hear about quality scores or so that either the lab gives you information and says, hey, here's the sequence and the quality of this one is lower than the other one, so use the data with more caution? Or how often do you go back to the lab and ask about quality scores of a specific sequence? Like if you have one patient where you like, that should really, epi indicates it's part of that patient is part of an outbreak, but there are like too many SNP differences. I wonder whether it's a lower quality sequence. Does that ever happen? Has that ever happened to you, where quality scores come up in sort of the practical interaction between epi and lab?
So I would say that it's not necessarily on a case-by-case basis, but what does happen is as we're waiting for analysis, there might need to be a rerun of a specimen because the quality score was lower. And I never really understood what that meant until I started to go through the material this past year and understand that there's a number of quality scores that they need to meet before they consider it analyzable.
Okay,
and
yeah,
so
that makes sense.
It might impact turnaround time.
Yeah, that's what I expect. Say, hey, you want this genome sequence, but you have to wait another 5 days because in the first run it came back with a low quality score. Okay, so we got a couple of questions coming in from the audience, and I'll read them to you, and we can sort of tag team to answer them as whatever will work for you. So the first one is, how do you know if the sequence you're looking at is from a plasmid, a bacteriophage, or the main gene? So if you want to answer them, or if you want to punt it back to me, I can try to give it a back too.
I will definitely punt that one back to you.
So there's different approaches for that. The most common one really goes back to the reference genome, where you already know something about an organism and you already have a database that will help you to determine whether this given sequence is a plasmid, a bacteriophage, or a gene that is found in all organisms. In addition, there are some other tools that you can look at the specific sequences you find. So sometimes when you have a plasmid, it's found in multiple copies. So one genome, a bacterial cell might have one genome and 100 copies of the plasmid. So you can see that in terms of the reads, whether it's a plasmid. And then sometimes you can see plasmids have specific sequences on them that allow these plasmids to replicate independently of the genome, or that allow the plasmids to move from one bacterial cell to the other. So if you find these sort of signature sequences, you can often say that it is a plasmid. But then there's a situation where you really can't be 100% sure. So it works in a good chunk of cases, but not in all of them. Another related questions was wondering how are the mobile genetic elements taken into account in the analysis. So the simple sort of short answer is really often they're taken out in the analysis because they confuse the analysis. And I can provide you with a practical example. So we sequenced that whole genome sequencing of 2 Listeria monocytogenes that came from the same food processing facility 12 years apart. They looked similar in PFGE, so we were wondering, are they actually the same Listeria or are they a different one? We did the initial analysis and they were different by 3,000 SNPs. We're like, well, okay, they're pretty different. But then we looked at the analysis some more and realized that there were some mobile elements in there. We took out the mobile elements. In this case, it was a phage, a prophage. There's a phage, a virus that hopped into the genome. Once we took it out, the 2 were different by 1 SNP. So it completely changed the conclusion from us thinking, wow, they were very different, to once we took it out and analyzed it without it, we could realize that they were actually very, very similar. So if you want to just look at relatedness, often they're taken out. But then if you want to get information like, is the organism antimicrobial resistant or not, you have to use them, because some of these important genes, like Madhu mentioned, antimicrobial resistance genes are often found on these mobile genetic elements. So it depends on your question. If you look just at the relatedness, they're typically taken out. If you're looking for relevant phenotypic and bacterial characteristics that might help you judge how severe an outbreak is, whether it's antimicrobial resistant strain or not, you will analyze them. But typically you keep analyze them separately from that relatedness analysis. And those of you with the questions, if you answer the questions and you have some follow-up questions, just type them in. Here's another one. Will you please give an example of how you see this impacting an outbreak investigation? And I presume the question was around mobile elements. So Madhu, have you ever seen, do you have an example from your experience where, presence of mobile elements or how they were analyzed really affected how you use data or used information?
Oh, I'm trying to think. Right off the top of my head, I'm not coming up with one, but let me think about it for a second.
Yeah, as I said, the thing I can imagine is another example is where it's not whole genome sequencing alone, but it was a listeriosis outbreak in Canada where they found, and this happened over and over again actually, where you find multiple PFGE types and multiple isolates that look like they're different strains by other methods of DNA fingerprinting or subtyping. And often these differences can occur because of mobile elements or plasmids. So even you could look very simplistically, you have a human isolate, isolates from multiple humans, same serotype, look like linked to the same source, but some are resistant to antibiotics, some are not. You could think, well, they're very different, they're resistant or not. But then if you actually do the genome sequencing, you might realize they're exactly the same except for presence, absence of a plasmid. So they're more closely related than you thought they are. So often mobile elements can make you think two organisms are different when they are actually different, but the difference happened very, very quickly so that they could still be linked to the same source. Okay, any other questions there? I have another one, Madhu, about around the example you gave about, I interpreted that seeing that your lab switched from MySeq to a HiSeq or NextSeq, where it meant suddenly more isolates were required before they would run them. Have you ever seen in these cases where you understand the workflow in the lab, but you have sort of a high priority set of isolates that need to be sequenced? Do those interactions happen? They say, I know you guys are waiting for 96 isolates, but this is really important for X, Y, and Z reason. Can you run it sooner and don't fill up a run, even if it costs more? How do those interactions happen?
Yeah, so actually we're in constant communication, and actually we just met this past week and talked a little bit about some of those thresholds is that our algorithms have changed a little bit within the past several months. And I think, what we're thinking of now is, if there was something that we really needed much more quickly, yes, I think they'd be able to run it on a MySeq. At this point right now, Wadsworth is running all Salmonella, so there's also a difference between running and getting a sequence and analyzing the data. And so they're going ahead and running all the sequences, but for the ones that are for Salmonella, but for the ones that we have as high priority, they'll end up looking for the analysis on that first rather than searching for clusters for us potentially.
Thank you. Yeah, so what I hear you saying really is that sort of interaction back and forth between the lab and Epi really is becoming more important as we apply whole genome sequencing and next-gen sequencing in some applications, just to really have an understanding even, are we at 94 isolates and almost at 96, or do we have one? Do we need to help you prioritize DNA sequence analysis, etc.? Do you have any other examples of how your interactions between the epidemiologist and the lab have changed as you're implementing whole genome sequencing, and what are some of the potentially the other important ways to interact more?
Yeah, I would say with just the epidemiology team we have now and looking at cluster analysis, they've really started to refine how information is shared. Before information was shared via a lot of emails. I think our Amy Robbins, one of our epidemiologists, is really on top of a lot of the clusters, and we've shifted into a common platform, a SharePoint platform, to be able to share data. So even just communication via electronic means has changed between us.
Okay, so it's not just talking to them, but also setting up some of these other infrastructures. So we had another question that was, can you explain how Res Finder works? And Madhu, I don't remember whether you had that on one of your slides or not, because I can't remember what it is actually.
I don't think that I had Res Finder.
Do you know what it is?
You have something on your screen. Do you want me to stop sharing?
Yeah, you can take a look at it. I don't remember what it is. So if you can find it out, or if the person who asked the question maybe can provide some
yep, I can do that.
Clarification what Res Finder is and what he or she is after in that question to help us answer that question. So if we can't answer it here, we will do search it and answer it on our website. So we will also post answers later on. While we're waiting to sort of get some clarification there, or maybe get some other questions, just a quick note that we have a survey regarding this webinar that we just emailed out. We just chatted out the link. So if you want to take that, it will help us to sort of continuously refine these, and also help us as we plan for the next webinar to make sure it's as useful as possible for everyone. With that, asking for any other questions that someone might have. Here's ResFinder as an online resource to look for AMR genes. Okay, so how these tools work, and so I don't know ResFinder specific, but I think I can provide some sort of insight of how they generally work. These tools will virtually always rely on a good database. So what you have a database of all antimicrobial resistance genes that are known, and then when you sequence a genome, you use the data from that whole genome sequence and search it against that database of all antimicrobial resistance genes, and to define whether a antimicrobial resistance gene is found in the genome you sequenced. So take you isolate salmonella from a patient, you sequence the whole genome sequence, you search it against the antimicrobial resistance database, you find a beta-lactamase gene, then you can say this organism should be resistant to penicillin and certain other beta-lactams, depending on what specific beta-lactam gene you find, for example. Very similar to whole genome MLST, from a bioinformatics perspective, there are two different approaches with these systems. One of them is that you actually don't assemble the genome. So you don't go through this process of assembling the song, but you just use the small fragments that come straight off the sequencing machine and map those against this database to see whether antimicrobial resistance gene is there or not. Or you can run this on an assembled genome, so where you create the contigs and then search against the database. So most data suggest that these systems are actually, particularly for organisms where we understand antimicrobial resistance well, can fairly well predict the phenotypic resistance of an organism. We've done some work, others have done and published work, is if these databases predict that a given salmonella, a given tuberculosis, Mycobacterium tuberculosis is resistant to an antibiotic, it typically truly is resistant. So these are very, very powerful tools. Very good question. Another question is, what is the turnaround time for PFGE results compared to sequence results? And do you think turnaround time will continue to improve for sequence results? What time frame do you think we'll reach for sequence results in the next few years? Madhu, maybe you can comment on what turnaround times you're getting right now, and then what do you expect to see in the future?
Yeah, I think it depends where things are in the workflow. With moving over to trying to fit 96 specimens, right now, because it's not peak season, sometimes it can be about 3 weeks. So it is longer than us getting PFGE results. I think we're hoping and anticipating as it starts to become busier. You know, this is a conversation we've had with our colleagues over at Wadsworth that, you know, as the season starts to pick up, those results will probably, the timeline will start to shorten from you know, to closer to a week or two. You know, I'm definitely a novice as far as knowing, you know, what the potential might be for obtaining sequence results in the next few years. You know, I wish I had my crystal ball as well, but I think, you know, as things get more and more refined, that, you know, initially it might take a little bit longer than it's been taking for PFG in some instances, but I think it'll start to get better within the next few years as more and more moves over to whole genome sequencing.
Yeah, and to just quickly add, I think in a perfect world, if everything aligns, all the stars align, and you have an isolate and you can get it immediately on the sequencer because it's isolate number 96 as you're waiting for these 96, so it's isolate number 24 as you're waiting to get 24 isolates for MySeq. From putting it on the seq, if you're at that stage, it can be as little as 5 to 7 days, really. So in those situations, it is almost competitive in terms of turnaround time with PFGE.
And I'll add, for the ones that we request as high priority, that is about the turnaround time, about 5 to 7 days. It's fairly quick.
Okay. Another question was, will there be a system like PulseNet for whole genome sequencing? Maybe GnomeNet. Nice try there, Willie. Madhu, any thoughts on that, what that system will look like?
I mean, I think that they are starting to move into bioinformatics to be able to analyze the data and essentially upload to, like, NCBI is essentially what that system is going to be like, you know, the repository for the sequences. So I do think it is going in that direction. I think it's more the nomenclature now associated with it, which I'm hearing that they're starting to develop a zip code so we can start to communicate in a standard language across the nation, but I think it's moving in that direction.
And we'll cover a little bit more of that in the next webinar when we talk about whole genome MLST data. So I think GnomeNet actually isn't that far off, because as we specifically talk about foodborne pathogens, really the PulseNet software, which is bionumerics, is being updated and modified and further developed to be able to deal with whole genome data in the context of whole genome MLST. So it really will be the same software platform, the same tools used, but now rather than feeding in pulse PFGE patterns, we will feed in whole genome sequence data, and we will get back what Madhu mentioned, zip code type, where it says, like, this is type 12211, and then if you have two isolates that have the same type, then you know they're identical. So that's definitely what, on the public health side, those of who work with foodborne pathogens, what you will see. For other organisms, whether that's viral pathogens, mycobacterium, tuberculosis, those might be different types of platforms that are seen, but on the foodborne route, that's what we likely see. So with that, it is 3:58. I think we actually got through most of our questions, and if nothing else comes up, we have a cute comment from Willie, who was suggesting a mascot for GnomeNet, and then mascot obviously will be a gnome. Very nice. We have one other question which we're going to try to quickly. Final question is, how far are we from reducing the minimum threshold of 96 per run to a smaller number per run or individualized runs? With a MySeq, it's more like 24, so 96 is really for certain platforms. I think we're moving very, very quickly to having the ability, a full run being a smaller run and being cost competitive. There's some alternative technologies that are being developed. So I could see the possibility of running really anywhere from 1 to 6 isolates economically. We can always run 1 isolates, but it just costs me as much as running 24. today, but in the future, I think that is getting fairly close. And so I think in the long term, this is not going to be a bottleneck. So with that, thank you, Madhu. Thank you, everyone, for dialing in. And I mentioned again, we will post the webinar, we post the slides, and hopefully we'll see some, if not all of you, at our next webinar. Thank you very much, and goodbye.